The title for this podcast is Molecular Inheritance. The molecule of inheritance obviously being DNA. So we're going to go through a review of DNA structure, how it works, how it makes proteins, all of these things which we talked about in biology, but you probably need a little refresher. And then also a couple of additional things that we didn't talk about when you took 10th grade biology. The first topic I'd like to discuss is a couple of the experiments that were done to actually discover the structure of DNA. For a long time, scientists didn't know that DNA was the molecule of inheritance. In fact, they really didn't even know what DNA was. They knew it existed, but they didn't know what it looked like, the chemicals it contained, and things like that. In fact, they were pretty convinced that proteins were the molecule of inheritance. In other words, it was the proteins that carried the instructions to make us who we were. So they did some different experiments to kind of get at what that might be and how that might work. One of the first experiments that kind of led us down to the path of DNA being the molecule of inheritance was done by a guy whose last name was Griffiths. And what he was working with was bacteria that could kill mice. And in this particular type of bacteria that he was working with, he did a series of experiments to see how that would work. Some of the bacteria could kill the mice, some of the bacteria didn't kill the mice, and he was trying to get at what that was, what happened. So he did a series of tests, as I said. In one of his first tests, which is represented here, he took one form of the bacteria, injected into mice, and they died. I think the diagram does a pretty good job of showing that. The second experiment, he took a related strain of bacteria, injected into the mice, and they lived. In the third experiment, he took this original DNA, or this original bacteria, here we'll call this bacteria A, and he killed it with heat. So he destroyed the bacteria essentially, so that's what this represents. And when bacteria A was killed with heat, the mouse lived. The final experiment he did is he put this heat-killed bacteria with bacteria B, which remembered also did not kill the mice. So in other words, the heat-shocked bacteria, which didn't kill the mice. The original bacteria that didn't kill the mice, so these two added together, B plus C, is this last one, and it killed the mouse. And you can imagine how surprised he was, because he took two bacteria that on their own did not affect the mouse, but together, when combined, they did affect the mouse. And even more surprising, when he actually took the bacteria out of the mouse and looked at it under a microscope, bacteria looked like type A, that virulent type. Virulent is the word they use to describe the bacteria that could kill something, so the infective type. So he proposed that there was something that was still in existence in these heat-killed bacteria that was transferred to these non-virulent or non-dangerous bacteria that allowed that bacteria to become dangerous once again. And this is that molecule of inheritance, whatever it happened to be. Again, they didn't know at this point what it was, but this was the first clue that there was something inside those cells that could be transferred back and forth that allowed these traits to be inherited. The next experiment that was done that tried to narrow down what this molecule was done by a couple of scientists. Last name of the first guy was Hershey, I like the chocolate bar, and the last name of the woman involved in the experiments was Chase, Martha Chase. So Hershey and Chase. Now they were trying to separate out, was it a protein or was it something else in the cells that was this mode of inheritance or this molecule of inheritance. And they thought a great organism to use to test this out would be something called a bacteriophage. I'll just write that out for you so that it's easier to remember. Bacteriophage. What a bacteriophage is it's actually a virus. That's what that phage part means. And it's a virus that kind of looks like a to me anyway, like a lunar lander or something. So it's got these little parts on it that attach to the cell, to the outside of the cell, and this body part in the middle, if you want to think about it like that, is made of protein and DNA. So the container part itself is the protein, and then inside the protein is the DNA. So they thought, well, if we want to figure out if it's protein or DNA that's changing these cells, let's test with these bacteriophages, which have a structure that's both protein and then a second structure that's DNA. So what they did is they took some of the bacteriophages, and you can see them up here infecting a bacterial cell. So they took some of these viruses and they basically grew them in a particular chemical, okay, which was actually, I think, a sulfur or something that had been, the important part is it had been exposed to radiation. So when the bacteria or the viruses grew, you can see these little viruses again on the bacteria, they now look pink. So they used that radioactive sulfur to build their protein structures. So on these little guys, again, here's one little bacteriophage. You can see they're pink, like the radioactive sulfur was. So they have now radioactive protein in them. Their structures are made out of radioactive protein. They then took a second set of bacteriophages and they grew them in a radioactive phosphorus. Phosphorus is in DNA, so the DNA portions of the bacteriophage were now radioactive in that group. And you can see the DNA inside there is blue. So again, in the first group, the protein structures of the virus were labeled with this radioactive chemical. And in the second group, the DNA portions were labeled with a radioactive chemical. So then they let them infect the host cell. That's this kind of bean-looking thing here. That's supposed to be a bacterial cell. So they let them infect them. And again, they didn't know if it was going to be putting DNA or protein into this host cell. They didn't know what the molecule of inheritance was. Step 2, they kind of mixed it all up together to see what was inside the cells, essentially. Step 3, down here at the end, they let everything settle out. Down in the bottom of the solution is the actual cells themselves. They're more dense, so they sink down to the bottom. And at the top of the solution is the leftover parts of the virus. Again, here as well, down at the bottom are the cells themselves, the bacterial cells, and at the top, the leftover parts of the virus. Again, just settling out by density. What they found was the DNA, or the information that went into the cells, okay, was labeled with that phosphorus. Okay, so down here, in this first picture here, you can see down at the bottom, the stuff in the cells is blue, and that means it's the DNA that's in the cells. Whereas in the top picture up here, the stuff above the cells is pink. So that means the viruses themselves have the protein. So in other words, they were able to figure out that the viruses injected the DNA into the cells and the protein itself was not injected. So that gave them their clue that it was the DNA and not the protein that was that molecule of inheritance, the thing that was carrying along the genetic information. All right, so now they knew what DNA or what molecule was the molecule of inheritance. They knew that DNA was responsible for passing on information. But like I said, they didn't know really anything about DNA because nobody had thought it was very interesting before this. So we had a couple more scientists work together with information from various sources to discover what the DNA actual structure was.